Thank you, Carol, for organizing this debate. And it is a debate, and thank you for the offer of truce, Ben, but I was forced to pick a side. So just to convince you that I'm serious about this, I'm wearing a suit and tie, which will immediately come off after this presentation. I'm also using my full titles. I'm a professor of urology and molecular biopharmacology. I have nothing to disclose. And I thought the best way to prove the case of the importance of smooth muscle in the development of ED is to look at the treatments that are used in the treatment of ED. So I'm going to discuss some of the established treatments and some of the preclinical treatments, the ones particularly that come from my lab. I'm going to talk about PDE5 inhibitors, MaxiK gene therapy, topical delivery of nitric oxide, and combination therapy of nitric oxide and PDE5 and alprostadil. So similar to Trinity, I have a seesaw representation. This is to convince you the correct regulation of smooth muscle tone is central to erectile function. So smooth muscle regulates the flow of blood into the corporal tissue. In normal tone, when less blood flows into the penile tissue, you have flaccidity. If that goes too far and you have heightened contraction of the smooth muscle, then this leads to erectile dysfunction. The opposite side of the coin is when you have reduced tone, you get more blood flow into the penis, you get an erection. If that goes too far, you get priapism. So I call this the Goldilocks of erectile function. Not too much contraction, not too little contraction, but it's the smooth muscle that regulates erectile function. I just want to give a very simplified overview of the steps that lead to an erection. Nerve stimulus results in activation of nitric oxide synthase, which is then amplified in the endothelial cells. And then this, following the stimulation by the nerve, then the mechanical effect is produced by the smooth muscle tissue. Nitric oxide activates cyclic GMP cyclase. Cyclic GMP then in turn activates protein kinases, affects ion channels, and normally calcium levels are maintained high in smooth muscle tissue, and this results in the flaccidity contraction of the smooth muscle tissue. But activation of these ion channels leads to lower calcium levels, and the myosin light chain relaxes, smooth muscle relaxes, and then neighboring cells, the signaling molecules are transmitted to neighboring cells, and this results in relaxation of all the concerted relaxation of the cells within the penile tissue, and you get an erection. So oral PDE5, we've heard about them in significant detail today. We did hear that they are expressed not just in smooth muscle tissue, but they are predominantly expressed in smooth muscle tissue. So their effect is to prevent the breakdown of cyclic GMP and cause a heightened relaxation of the smooth muscle tissue, which then allows an erection to develop. You have to have a neuronal signal to start this process, so you do need neuronal input. So you could argue that these PDE5 inhibitors do rely on the activation of guanylate cyclase by nitric oxide. So nerve function is critical to the action of PDE5 inhibitors. So as we heard, patients that undergo prostatectomy or diabetes, where the nerves are damaged, PDE5 inhibitors are not effective. So going to a treatment that we developed in our laboratory back in the early 2000s, we had shown that the MaxiK, a potassium channel, plays an important role in erectile function. We showed in animal models by our group and others that the MaxiK is involved in relaxation of smooth muscle and can induce improved erectile responses in animal models. We have used aging models, rat models of ED. When we injected a plasmid expressing MaxiK into these tissues, they resulted in improved erectile function. This was the basis of the MaxiK gene therapy, which has been used to treat ED, and more recently, bladder dysfunction, which is a smooth muscle disorder. And these are the only treatments for a benign urologic disease that have actually gone into phase 1 and phase 2 clinical trials. In 2006, it was featured on the cover of Human Gene Therapy, and you can see one of my early attempts of using PowerPoint to make a model for these effects, and I'm still at that level of PowerPoint presentations. So, you could argue that MaxiK activity relies again on nitric oxide release from nerves. So again, nerve function is critical. MaxiK is expressed both in neuronal and smooth muscle tissues, so treating corporal tissue with this gene might allow it to be expressed both in neuronal and smooth muscle tissue. Just one argument against it being effective in neuronal tissue is that we had it expressed from a smooth muscle specific promoter and showed that could enhance erectile function in atherosclerotic cynomolgus monkeys. So more recent work from our lab have explored the use of nitric oxide nanoparticles, which can topically deliver nitric oxide to penile tissue. So this is applied to the penis of rats. The nitric oxide nanoparticles, when they get into an aqueous environment, release nitric oxide. So when they're applied to the animal, nitric oxide is released, and that initiates the molecular signals that initiate a relaxation of the smooth muscle. So this circumvents the need for neuronal stimulation. So to show this in practice, we used a rat model of nerve injury, which is a commonly used model for ED resulting from radical prostatectomy. We bilaterally transected the cavernous nerve, waited 1 week, and then we applied these nitric oxide nanoparticles, and we used cavernosometry to show that when we applied these nitric oxide nanoparticles, we could elicit an erectile response. And we went on to combine this topical delivery of nitric oxide with an oral PDE5 inhibitor. We've done it for several, which I'll show in the next slide. So circumventing the need for nitric oxide from the nerve endings, and then adding PDE5 as orally increases the number of erectile responses over time. It also both increases the number of erectile responses over time and the time for onset of the erectile responses. We've recently gone on to show that the effect is synergistic with all of the FDA-approved PDE5 inhibitors. So nitric oxide acts synergistically with PDE5 to elicit erections in a rat model where there is no nerve stimulation. And the final example of a drug that acts directly on smooth muscle is alprostadil. prostaglandin E1, it activates adenylate cyclase and activates then the downstream pathways that lead to smooth muscle relaxation. So this, again, does not need neuronal input. So, this is my debate conclusion. Smooth muscle provides the mechanics for erectile function. The only FDA pharmacologic treatments for ED target smooth muscle function, and the majority of preclinical research for ED treatments target smooth muscle. And the reason I believe for this is that smooth muscle has unique biochemistry. So, for example, the PD5 expression is located in smooth muscle tissue. Nerves, on the other hand, don't have unique targets that can be used pharmacologically. And also, as I've demonstrated with the use of the nitric oxide nanoparticles, as an external organ, penile smooth muscle is more accessible than internal organs. So you can actually get pharmacologics targeted to cavernosal tissue. But really, I have to agree with Ben. This debate should be a draw. For one thing, I have an R01 grant looking at the role of nerve regeneration in erectile dysfunction following radical prostatectomy. So at that point, I had to convince them that neuronal function was essential for erectile function. Also, I think looking at tissues in isolation is a mistake. Looking at cells in isolation is a mistake. And we need to take a more holistic approach to understanding erectile function. Looking at both the smooth muscle, which provides the mechanics, the nervous system that initiates an erection, the epithelium, and connective tissue. So I think, the conclusion is that I hope this debate is a draw, and that we recognize that all of these tissues have to act in concert to produce an erection, and conversely, any of them can go wrong to result in erectile dysfunction. Thank you.